Thank you for the introduction. So for today's talk, I'm going to share some of my recent work since I moved to Hawaii. And a lot of work we're getting here is mostly preliminary results. But the results is like quite interesting and they generate some like interesting question I want to share with you. And to this group of people, I think, it would be interesting with the microbe stuff. So to me, I'm in training in soil physics, and in this topic, I'm going to talk about micro, but it's going to be from the soil physical perspective, So, but the talk is going to be at the intersection between microbiology, physical, and hydrological processes. Hopefully, at the end of this talk, I will convince you the importance of thinking about the physical habitat of the soil and how does that culture the fungi behavior. Okay. All right. In the very beginning, I want to acknowledge my collaborator and the students who have done this work. Most of the work is done by this student Ishwara, and there's some and also some work from the Alec is a high school student from Hawaii, and then this work is a collaborative effort with Dr. Movin, working on with fungi. And we also get some like funding from the Core Box and also the Summer Research Institute. And there's this like microscope facilities supporting our work as well. Okay. All right. So the outline of this talk today, I'm gonna start with like my journey exploring soil science and soil biophysics, and to give you some idea where do I come from, and to many of you don't know me, and also talking about what are the topics we're developing in this lab, soil physics lab at UHM, and then later on, I'll jump into this topic talking about soil as a living habitat, and this is more like my personal view on this, like how soil is being a habitat for these microbes, and also how these microbes are actually distributed into these microhabitats and so on. And then the third topic I will talk about, the recent work we've been doing with Dr. Nguyen on this more like a mechanistic study on the soil biophysical controls of fungi growth, and also how it affects the soil evaporation. And we're using this Hawaiian clay soil as an example for now. And then the last, I will, of course, I will talk about preliminary results and then discuss these results, and then a lot of like things we don't understand and generate some like new questions. And the end, I will leave some time for question and discussion. Okay. All right. So this is, I want to start with my journey in soil science. So when I started as a grad student, I was really into this soil processes and then how soil control water and air, and also the nutrients to plants. And also by the time when I started my PhD, the nanotechnology is a really big thing. So I started to try to carve something for myself to look at how this nano stuff is moving in soil. So what I first studied is soil colloids, which are the not very small particles in soil, and how these nano-sized particles are actually moving through the soil and release, and how these nano-sized particles can carry stuff like nutrients and carbon, and then affect like a subsurface flow and also the adjacent streams. And then later on, after I finished my PhD, but before that, my work has nothing to do with biology. So I want to mention that. So after I finished my PhD, I started to work on another adventure. This adventure is super different from what I did at the very beginning. I work on rhizosphere water and nutrients dynamics. And so essentially, we try to understand how roots are extracting water and nutrients in the rhizosphere to know, how can we learn this process to develop some like an efficiency in nutrients management plan. So at that point, I learned plants can be really amazing. They can actually modify the soil in the rhizosphere and actually control these flow processes. They can actually affect the efficiency of their using this water and nutrients in very specially heterogeneous water nutrients distribution. And then after I finished this postdoc experience, I started to work on more on the microbe side. Like this microbe side, we work on this the bacteria called plant growth promoting bacteria. So it's basically try to use this bacteria as a biofertilizer to improve these plants, especially in how does that mitigate the salinization problem in soil. So all these processes help me to have a better appreciation of this biology, how this biology can affect the physical processes. And in the end, I realized, all these things can be unified with some like a series, basically the transport behavior of these key soil constituents. And so we studied I studied different particles, solutes, and also microbes. All these things are actually carried by water and nutrients, but also they can actually provide some feedback effects and control the soil as well. So my overall thinking is, after that, is thinking to building this soil sustainability through the better understanding of this soil biophysics. So that's the overarching goal of my research, for the lab where we're developing now. Okay, so these are three major topics we're trying to work on after I moved to UHM. So first thing is looking at this soil hydrological property of tropical soil, this landscape scales. And so basically looking at how these hydrological properties vary with contrasting environmental settings, especially the mineralogy, vegetation, and management. And can we predict these hydrological properties from basic soil properties? Because these are really expensive measured properties. And then if we can do some estimation from the other properties, then we can use it for the bigger community, to help supply this to help the water management across the landscape. The second question we're trying to address is this food water energy nexus in agro ecosystems, and this is at a plot and profile scale. The idea is try to understand this complex plant system like agroforestry, which you have multiple type of plants that can self-regulated water and energy use. And how can we learn this system and then redesign a more efficient cropping system? That's the question we're trying to ask. And then the third topic is this even smaller scale, is working on this soil microbe and their interaction with physical habitats. And this will be mainly on the core and pore scales, and because the microbes are usually stay in soil pores. And the major question we try to ask is, how do microbes respond to these physical conditions and modify this soil for beneficial functions like water conservation? Okay, so for today's talk, I'm gonna mainly focus on this third topic. Okay, all right. Starting this talk, I want to first mention today is the Earth Day. Today is the Earth Day, and then the key theme of the Earth Day of this year is our power, our planet. Okay, so it's a nice chance, and here is a nice chance to mention the power of soil, right? So this is you guys might already heard about that. There are more organisms in 1 teaspoon of healthy soil than there are people on Earth. So it's like even you hold this very tiny bit of handful of soil, you are actually holding a lot of life inside of this soil, right? So this is not just like a code. This is the results. You can from this paper published by Taken and Orr from 2017. They look at the genome size of different environments. We collect this the soil and rhizoplant, marine, freshwater, and digestive stress. So they look at this. These are the soil. Soil microbes carries a large gene rich genomes than microbes than aquatic and aquatic environments, right? So why is that? That's the question. Why is that? Because the soil is actually holding a really complicated, environment or microhabitats due to this physical environment, and this create different type of pocket of the environment. They can actually generate different living conditions, so different microbes can actually live in the same area of this very tiny bit of handful soil. Okay, so but there are a couple arguments about this topic. When we talk about this idea of this biodiversity in soil, we often lack of talking about the habitat, and then there's some interesting paper talking about the search of meaning of the life in soil, talking about how does this physical habitat can actually affect the distribution of this soil microbial community and then activities. But also there another thing is this microbe can also have some modification effect to generate some feedback loops. So this is another paper talking about the interplay between the soil microbial community and soil properties. So essentially, this is a chicken and egg question. Soil is a living habitat, but these microbes can also generate some interaction that can modify this soil environment and to make them more habitable. But there's a lot of unknown behind this. We don't know, how this physical function affect the soil microbes, but we also don't know how this interplays is happening in the soil. So to take back a little bit about this the size what's happening in the soil, I want to show you guys a size spectrum of different components of the soil. And then there's a biological component. There's also this chemical or physical components. And then the key physical component, often when we talk about soil, is clay, silt, and sand. And of course, you guys know more about than me. These are the different type of microbes and different size. And the interesting part is this is the diagram of key soil components: clay, silt, and sand. Sand is pretty big, and then silt is smaller. Clay is pretty small, and bacteria is in between of this clay and silt. But virus is in this 1 nanometer to 1 micron size, and the algae and fungi for the cross section area is 5 to 10 micron diameter, but can be meters long. So essentially, these microbes need to find their way to attach to these particles or find out void space to live within this microhabitat, which I will talk more. Okay. So when you go outside and then you take a scoop of soil, and then you just take a look at a very small of these fragments of these particles, and then if you can often see this type of a structure is called soil aggregates. And this aggregates, if you smear it or you break it, it is actually composed of these unit particles like clay, silt, and sand. But if you put them under this microscope, what does it look like? It looks like they form like a castle, all these particles are formed together. And these are mostly, these are something also with organic matter, and also they form this type of structure that actually affect, the pores and also the particle arrangement. So if you look at what's inside of these aggregates, and this is the aggregates from the literature. They have done this X-ray tomography, and then you can look at X-ray tomography at this single slice, and then you can look at the pores were inside of this aggregates. So this is the 3D regeneration of these pores. And the red means the narrowest pores, the blue, the green means a wider pores, and then this white curves. If you look at these aggregates, this is from the one side to the other side. this is the shortest pathway if you want to move through this pore to the other one. Okay, so so what does it mean? So it means, you know, like this arrangement of these particles create these aggregates, but these aggregates are actually porous, and this porous stuff can create this type of tunnel result, a tunnel like a formation architecture, which can affect the microbe distribution, because the microbe has to live in these pore spaces. And then, you know, think about it, if you were a microbe, you have to walk through this whole you know, pathways from this side to this side. There's no highway, but you have to work like a you know, back and forth and go through this tunnel. So the soil structure is actually is basically talking about the arrangement of these particles. And this structure is actually affected not just the microbe distribution, but also the resource distribution, like oxygen, water, and nutrients. And these are controlled by this pore network. Okay. Okay. So if you look at these microbes or bacteria and then the fungi, this is often, you know, what you look at. You know, this is the minerals, and these are the fungi, and these are some like a I think extracellular polymer substance, and then these are fungi, and between the aggregates, you know, pay attention to this number of the size of these particles. This is 2 micron size particles. This is 5 micron size of aggregates. So these bacteria and fungi are actually, you know, tapping in different type of pores, you know, and in this case, okay, which means the habitat can actually affect where do they grow. Okay. So essentially for microbe, it is more like within this very tiny bit aggregate, they're actually searching their paths through these three-dimensional soil mates, right? They have to go through this pathway to figure out their resources to live. This is and then all these pore space are not equally are not equal, because, you know, some of the pores are small, some of the pores are large, and the smaller pores are actually can have different effects in terms of this capillarity. So if you look at in terms of water retention, so if you look at this experiment, it's very simple. So you have this tube with larger diameter, and then on the left side you have a smaller diameter of these capillary tubes, and then you measure this capillary rise. So this water movement is actually higher in this smaller pore radius, with the smaller pores with smaller radius. And then this is due to the capillary force, and then this is due to this upward capillary force due to interfacial tension. And then under equilibrium, it's going to be balanced by these downward gravitational forces. Okay, so and then this can be estimated. The capillary pressure can be estimated due to using this equation, which is relationship, which is as a function of the surface tension, and then the contact angle, which is like the angle of this film is attaching to this solid surface, and then the radius of this pore. Okay, and then this capillary pressure is one of the key drivers that affect water retention and infiltration and evaporation, which I will get back later to talk about this in the when we talk about the fungi experiment. Okay. But in soil, if you think about soil as a bundle of these capillary tubes, then you have all kinds of pores, right? So this is a very typical experiment of this pore size distribution of typical sandy loam soil. And then you can see this is a frequency of different types of pores. And then this is probably due to different type of compaction, I think. You know, so it's shifting this like the pore size distribution. And because of this pore size distribution of difference, and of course I showed you guys this capillary rise, and this affects this water hydration status in this type of like a pore sand and fine sand, because the pore sand has less capillary pressure, so the water distribution is going to be in this case is raising up pretty low elevation. But for fine sand, it can actually fill with all these type of pores. Okay. So this soil texture is tightly linked to this like a pore architecture, and this hydration status is also dependent on this pore size, shape, and pore viscosities. Okay. So what does it mean in terms of like a microbes? And there's some like a very interesting work at an earlier time from colleagues working on this type of system, and they look at, if you can control the rough surface and then make the rough surface like a wetting or wetter condition or drier condition, they use two type of microbes. One is like more flangile type of microbes, and the other one is less. So if you put them under very wet conditions, this flangile microbe will spreading across this whole plate on the surface, which all compete these like non-flangile microbes. But if you put it under drier condition, you can see this like non-flangile, the reddish color bacteria can be survived. So which means the habitat of this microbe habitat can be created due to this like fragmented water distribution within the soil, and then control the ecological community effect, community distribution of these microbes. And this is mainly due to the thickness of this water film controls this mobility of this bacteria. Decrease of this thickness of this water can actually increase a lot of viscous resistance, and surface tension pinning forces basically retain these microbes onto the surface of these rough surfaces. Okay. So I already tell you this is the habitat controlling the microbes, but these are some like recent study from us is we're working on this, like how this pore scale microbial control can actually affect the saltwater evaporation. So what we found is we use this plant growth promoting bacteria, which is the Bacillus subtilis bacteria, and then we put that into this evaporation system and looking at how does that affect evaporation. And what we found is this bacteria attached to this like contact line of this meniscus, which can affect the pinning of this contact line, which affect the water film configuration. This water film configuration change actually how water can move up through this capillarity movement. And so this affect, like this salt, the salt is also following this water flow. So affecting this, the salt accumulation within the pores due to this contact line pinning. But in controls, we don't have these bacteria. So this salt will moving towards to the surface due to this, like water can be more freely moving to the surface. So this result shows you at a microscopic scale, this bacteria can actually control this, the water film shape actually can have this really small scale process or mechanism can actually affect the larger scale evaporation processes. Okay. So digging into fungi, so these are some literature results, like how this pore architecture affect this fungi fungal habitat. And this is using the pore sand, and then they grow this fungi from this side, and then looking at how these hyphae are bridging this gap, and also looking at the distribution within this gradient from the immediate adjacent to this pore, the crack, and to this inner soil matrix. So you can see the hyphae length is actually decreased from the surface at here to the inner side of this soil, which means this pore size heterogeneity can actually affect this fungi growth as well. But the difference is like this fungi is not like a bacteria. They have to rely on the water film distribution, but this fungi they can actually grow by themselves, to look for some resources. So the scale they can change the soil is different from the bacteria. What I want to say. Okay. And then this is another evidence of how this soil drying can actually affect the colony radius and then the biomass density of this colony. And then what is measured potential is basically mean the soil is being dried. From here is wetter condition, it's drier condition. A soil drying from this wetter condition, the pores will be drained, until you reach more, until you have more empty pores where air can fill more empty pores. So in this case, you can see the mean colony size is actually increased as you increase the soil dryness. But the density of this biomass is actually decreased as you increase this soil dryness. Okay. So, but the system is mostly looking at a lot of system we're working on in the literature is working on this coarser materials. So basically sandy or coarser texture soils. And it is actually remains unclear how this fungi network develop in clay soils where most pores are inaccessible to fungi. Okay. So, but this is another literature results, from earlier time looking at this, how this fungi can actually reshape the clay soil movement. And this is they grow this fungi with this clay, I think it's clay kaolinite, and then you can look at the rearrangement of this clay around this hyphae. And also this is some other evidence showing you if you have fungi and then during this crack system you have more rough surface of this cracks, which can affect the water retention or gas diffusion sometime. So we really don't know how does this fungi network develop feedback mechanism that can affect the water flow behavior. So that's why my original thought we want to work on this. So let's jump into this specific study looking at this, how do soil fungi interact with, probably I hear myself from my computer. Yeah, so, okay. So how do soil fungi interact with the soil pore structure and affect soil water flow dynamics? Okay, that's the overarching question we're trying to address. So in this study, we try to address two objectives. The first one is how does this physical habitat affect this fungi growth? And so especially, we can examine this a pre-existing soil structure condition. How does that affect this fungi growth in this well-structured clay soil, which we can find in Hawaii? Which I will give you more explanation. What is this well-structured clay soil? And the second question is how does this fungi activities affect this soil evaporation process? And using this evaporation study. Okay. So this is the first part. This probably some of you have already seen this. This is oh, it's working. Okay, nice. Fine over there. Okay, so this is a really nice work done by our colleague Dr. Johnson, Dinik, and Taimas. They have shown the soil map of this whole Hawaii, and then I'm just showing this like a little piece of this, showing you the diversity of the soil. We have oxysoil, alluvisoil, vertisoil, then multisoil. They all have different type of mineralogy, and also the key thing is also the structured soil is the oxysoils, which we're going to talk about today. And this oxysoil is basically this reddish soil. Often you can see from the central Wahoo. This oxysoil, if you look at this soil by grabbing a handful of that, you can see this really develop the aggregates. And this soil is actually clay soil. And this clay soil, what is the unit particle is very fine particles, but they can actually form some aggregates. So these aggregates are forming this type of structure, like a sand size of particles aggregates. And within these aggregates, you have very small pores, but also between these aggregates, you have larger pores. Okay, and so oftentimes, this soil are considered as called as a pseudo sand because they have this aggregate formation. So when the soil is pretty wet, they behave like a sand because the water can flow through these bigger pores. But when the soil is become drier, the sand cannot retain any more water. No way, because the sand, all these bigger pores are gone, and the inside of this sand is like rigid, and there's no pore space. But within these aggregates, you have a lot of smaller pore spaces. So this creates a really unique property of this type of soil. It is often called bimodal, and this bimodal is often called, you have both interaggregate pores and also this. This is wrong, I think. You use the wrong. Interaggregate pores should be larger, and interaggregate pores is smaller. So, but anyway, they have both sides of the pores. Due to this, like the bimodal distribution of this type of like structure effect. Okay. So we can use this effect to use this like a well-developed structure to study how this fungi can grow into this type of soil. So what we actually designed is we use this C1 is controlled without inoculation of the fungi, and then T1 means the soil with the treated with the inoculation of the C is the control, T is the inoculation with the fungi, and 1 means the original soil with this like larger pores. And then 1.3 means we compact the soil to remove these larger pores. So which means this soil will only have this form. We minimize the larger pores, and most of the pores are going to be smaller pores within these aggregates. So essentially, what we got is these are the parameters. This is the bulk density, and this is the porosity of these pores after we compact. Of course, after we compact, the porosity decrease. And then this is the grammatical water content we use, the volumetric water content we use. Because we compact the soil, the pore space decreased, the volumetric water content actually increased. And this is also due to this compaction, the air-filled porosity also decreased due to this compaction. Okay, so this will, we artificially create a new structure, like to remove this like interaggregate pores due to this compaction. Okay, and then we add this like we sterilize the soil and then incubate it with this much of spores using the fusarium, and incubated at this like 35% of grammatical water content at 30 degree for 10 days. And my thought is like the compaction will decrease the interaggregate pores and the air-filled porosity, and resulting this lower fungal growth within the soil pores, but more growth above the surface under compaction condition. So if we look at the growth, we should see more of this fungi growth in pore space for this non-compacted system within the pores, but for the surface growth, we should see more growth on this compacted system, right? And then this is the result we got. As we expect, these are the controls where we have some probably indigenous species still growing, even we sterilize the soil. But this is the treated system with this like fungi, and then more of this compaction can actually affect more surface colony formation due to this compaction. Okay, and then the reason is likely due to this interaggregate pores is lost, and then air-filled porosity is decreasing. Okay. So a simple summary is soil structure and pore scale change can govern this fusarium growth. And then in this clay-rich oxysols, compaction change soil structure by collapsing these large pores and lowering this air-filled porosity. And then this fusarium actually shift from this internal colonization within the pores to surface dominating aerial growth. So the question to us is, how do these different fungi growth patterns physically influence saltwater conservation or evaporation processes? And then this is the next part of the talk, is looking at these fungi effects on saltwater evaporation processes. And then first, I want to mention, why evaporation? The evaporation is actually quite common and happening every day. And then evaporation consumes this 25% of incoming solar radiation, which is super important in terms of the energy balance of the world. And also this 60% of terrestrial precipitation returns back to atmosphere while this evaporation and plant transpiration. And then 20% is coming from evaporation. So it's really important in terms of this regulating this global water cycle. And for example, if we talk about the Oahu, this is literature results showing you there's this 720 million gallons of rainfall evaporating daily. And then it about twice of the island's water usage in 1990s. And 1990s when where we used to have used more water in the plantation. So right now we use even less water, so this is a huge amount of water actually controlled by evaporation. Okay. So that's why one of the reasons why we want to study evaporation processes. Another reason is more technical why we want to study evaporation, because the evaporation is a very unique process, but it can actually tell you how does the hydrological property change of the soil. Because evaporation rate change over time. If the surface is wet, then the rate is going to be flat. So because the capillarity will connect this surface to the deeper layer of the soil, so transferring this water to the surface. And the evaporation rate is mainly driven by this energy of this or weather condition of outside of the top surface of this soil. But later on, as the soil become drier, this capillary connectivity will be decreased because this breaking of this connectivity, and then the evaporation rate will change. So using this type of system, we can actually evaluate how does this capillarity, whether this capillarity can move this water up from the deeper layer to the surface layer. Okay. And also evaporation is very interesting, is as I mentioned, is associated with latent heat loss. So the change of this surface temperature can also tell us where does the evaporation happen. So which I will show you guys why is that useful. This is the experimental design. It's very simple. So we have this soil column, which is with control and also inoculated with this fungi. And then we put them on top of this balance to logging the mass loss over time. And then we have this thermal camera to looking at, this one of the treatment, which is the T1.3, so looking at the surface temperature change, try to look at, where does the evaporation come from. And then we also run this droplet penetration test, just trying to know, how does this water can be penetrating into the surface of the soil later. So the results is actually today, I can only share you guys this treatment because the other results is not really consistent. So I'm only showing you guys this compacted system. So this is a cumulative result, cumulative evaporation change over time. And this is the treated system with compact system. This is controlled with the compaction. The results is just showing you the evaporation actually decreased due to this fungi treatment. So which means, the fungi actually affect the water loss during this evaporation processes. And we can do further analysis of this data into more details. So this is the way we do is we calculate the evaporation rate, basically how fast of the evaporation loss during the day. And then the saturation level means, on the right side is the soil is become pretty wet. On the left side is the soil is drier. So what we see is not really totally clear, but it's really consistent result to see this stage change of this evaporation. So you can tell, from this stage 1 change to stage 2, but what happened is very interesting is the transition of this stage 1 to stage 2. For this fungi treated system, the transition comes at earlier time, which at a higher saturation level, which means the soil, although the soil is high, have a higher water content, but the capillarity cannot move this water up. So basically, the capillarity break, the connectivity break at the earlier time due to the fungi treatment. Okay. So so we want to understand why is that. So so this is why we do this droplet test, to see how fast of water can penetrating into these fungi surfaces. So we run this droplet test. It's very simple. It's just like having one small droplet, probably 50 microliter, and then just look at how fast of this water draining into the soil. So if the soil can generate a higher capillary suction, then the water should be immediately goes into the soil, right? But in this case, we see both of this treatment, the penetration time takes much longer for this fungi treatment. This is not really unexpected. It's actually funded in many literature. So this fungi network can actually affect the penetration of the water due to this wetting behavior. So this is what actually I think is happening. The capillary pressure is actually if you have a really high capillary pressure generating, so you will have some faster penetration of this water goes into these pores. If you have a lower capillary pressure between generation within these pores, then you will have slower penetration. So this fungal network may decrease this capillary pressure difference, and hence prolongs this penetration time. But so then what you want to look at, this equation, what actually control this capillary pressure is the surface tension, is the contact angle, is the radius of the pores. So these are the things everything can actually change this capillary pressure. So I don't know which one is controlling, so I'm listing everything at here. So some of this multiple physical mechanism can at play is this fungi can release these surfactants, which can lower the surfactant, lower the surface tension, and also secrete these hydrophobics and increase the surface roughness by forming a rough hypha network. And this can increase the contact angle. And then also this hypha enmeshment that can mechanically increase the aggregation and increase this inter-aggregates or reduce the intra-aggregates pore radius. So these are the things could happen, from the best biophysical side. This fungi can actually regulate this capillary pressure due to the change of different factors, which we don't have a way right now to test which one is more important. But there's even more interesting side of this is looking at the thermal graph of this soil evaporation. Like I mentioned, this evaporation is following this evaporation. When the evaporation happens, it takes heat. The lower temperature means actually higher evaporation happening. So this is the thermal camera results. Like you can see, at the beginning, the soil, the bare soil, actually contributing more of this evaporation. But later time, the camera actually tracking the lowest temperature value at this using this circle. So it's actually shifting to this location where you have a lot of fungi coverage. So which means evaporation initially occurs in soil with limited fungi coverage, and later transits to areas with greater fungi presence. But this is against what we're talking about. Just talk about using this capillary pressure model. So the capillary connectivity actually sustained in regions where this more fungi occurred. So I don't know what actually happened, but I'm thinking this might be due to this, this is like what we take some like SEM results after we finish the experiment, and then we see this type of structure on top of these clay particles. And then you see this fungi hyphae is attached to this surface, and also they form some like a network. And these can actually, if they are hydrophilic, then they can actually control some of the water retention. So these are some evidence from glass fibers, not, of course, not fungi hyphae, but these glass fiber, by rotating their angles, so you can see the water and can actually retain between these glass fibers in different forms. So my thinking is this capillary connectivity it can be sustained in some regions where this like fungi growth occurred. And then this is due to this core scale, this biophysical mechanism by this fungi network that increase this water retention. I'm thinking about that, but I don't know like that's true. So we need to find out a way to test it. Okay. So that's all I have. So this is a summary of the results I want to give you guys. So the effects of the soil structure condition on the soil is the high level of surface fungal growth, compact and non-compact treatment, likely due to this decrease of aggregate pores, and then this decrease of this air-filled porosity, which we will test it. We're currently testing this hypothesis now. And then the effects of fungi activity on soil evaporation process. This fungi inoculation can lower this evaporation under this compaction system. And then this decreased evaporation presumably contributed by the decrease of this capillary pressure induced by this fungi growth. But this is still question mark. We need to do more work to look at the fungi network. How does that affect the water? Okay. So the open question, like I mentioned, a lot of things we cannot answer for now. So, but these are questions we are trying to address. So, what type of biophysical mechanism are at play? For surfactant release, hydrophobicity development, or pore structure change? What are the pore scale regulation mechanisms? Does liquid retention increase at this hypho-hypho junction or hypho-soil interface? Or other mechanisms we didn't even talk about today, like fungi bacteria, fungi interactions, or viscous resistance by fungi network, or this EPS effect. So hopefully in the end, if we have a better understanding of all these processes, we can generate some solutions for bioengineering for agro-ecosystem or natural ecosystem management. Okay, that's pretty much I want to share. And I want to thank everyone for your attention, and I'm open for any questions. Thank you.